Hi, good evening, all of you. Hi, good evening, all of you. Hope I am visible to all of you and you are able to view the screen and hope you are able to get my voice. Please confirm. Hi, good evening, all of you. So, let us discuss about what we are going to discuss today, that is a continued process verification. So my name is Hitendra Kumar Shah. I am a CGMP compliance consultant, trainer and auditor from NADH plus GXP compliance services, and today we are going to discuss about the continued process verification. Now the question is why, the question is why we need to discuss this, because the point is I have audited so many companies till date. I am performing the third party audits, and during the audits, third party audits, gap audits, so many types of audits I have conducted. More than 500 sites I have visited. I found that most of the companies not implemented the continued process verification. Now you will say that we are performing the process validation in line with the traditional approach. Okay, but my point here is the process validation is now in a three-stage approach, right? Process design, correct? You remember process design, then process performance qualification, and then third is a continued process verification. So the point here is the process validation is in a three stages, and the third stage is missed by so many companies. And the companies who implemented this, basically, the companies who implemented this continued process verification, they are not also implemented properly, or you can say in line with the current guideline requirement. So I thought that I will discuss this to all of you. So meantime, you can just put a comment that whether I am audible to you and everything is okay, so that I can move further on the discussion. Okay. So this is just a brief about the today's training session. So please confirm on the chat box. Now what I want to discuss today is uh, the topic, and before discussing this topic I just want to put a good statement about the the requirements, the requirements given by this person, Theodore Levitt. Basically it is not related to any, the, you can say, the process validation and all, but the requirements, why I put this slide before discussing today, the he as per him, the people don't want to buy a quarter inch drill, they want a quarter inch hole. So the point here is we should understand the expectations. What is the requirement? People are not interested in the drilling machine. They don't want to buy a quarter inch drill. They want a quarter inch hole basically. Now why I am discussing this thing? Yeah, Mr. Ravi Modi, thank you for confirming. So what does it mean in our context as a process validation? Most of the people, they prepare a process validation protocol report, that's it, as a formality. So nobody is requiring you the process validation protocol and report. The expectations is whether you are able to manufacture the product in a consistent way or reproducible way, that you have to evaluate from the documentation or from the study what you are conducting. So the formal just a protocol and report, in a old traditional way will not solve the purpose. The expectation is something different now. Expectation is to evaluate and to prove that the process followed to manufacture the product is reproducible and you are able to produce the product of a consistent quality. You got my point. So this is just a brief about, or or you can say the introductory about this statement by Mr. Theodore Levitt. And I think we should start with this. So what we are going to discuss today, so this is the agenda for today's training session. So just now we have discussed about the importance of topics, and now we are going to discuss about the agenda. After this agenda we will discuss about the know your trainer. So it's a brief about the me, guideline requirements, specifically UGMP, FDA. Whenever I am using the terminology FDA, it means the USFDA and WHO. understanding the FD expectations about continued process verifications. So what are the FD expectations about the continued process verifications? Now the point is why I am focusing on here FDA. The reason is, I referred so many guidelines, EU, PEAKS, even WHO, FDA. I found that there is a very small content is given, or the expectations are in a very small way in all the other guidelines. But if you go through the FDA guideline on process validation, it is quite brief, quite detailed, and quite elaborated. And that is the reason I am more focusing on understanding of this topic by referring this FDA guideline for process continued process verification. Okay. Sampling considerations. So what are the sampling considerations we should consider? What type of statistical samplings and all, at least I will give some information about that. Then how practically we can implement, so practical view to implement we are going to discuss, where we will discuss some points. Each point we need to evaluate. Again, I am telling you, whatever I am going to discuss, it is exactly in line with the guideline requirement. Very clear. Okay, I am very clear about that. Okay, so that I am going to discuss today about the practical view of implementation. Then we will discuss about some differences between, brief differences between the EU and the FDA requirements for continued process verification and process validation. And after that, we will discuss about some learnings from the warning letters given by FDA. Okay, so many people, what they refer the warning letters to just go through the observations. But my point here is, when you are going through the warning letter about the observations, there are some recommendations or requirements are specified by the FDA in the warning letter, which also can be the basis for your understanding of the topic and further implementation of the topic. You're getting my point? So that type of learning also we are going to discuss through the warning letters, followed by the question and answers. So if you have any questions, please put your questions in the chat box. I will reply to your all questions. Okay, this is a live session, so that is the thing. And after that, vote of thanks and the contact details. So this is what we are going to discuss in today's discussion. Okay, so please put your questions in the chat box if you have, so that I can further take these questions during the discussions. So as per the agenda, a quick overview about me as a trainer. My name is Hitendra Kumar Shah. I am a CGMP compliance consultant, trainer and auditor from NADH plus GXP Compliance Services. I am having more than 23 years of experience, and this is a brief about my CV. I will not put more time here, but why I am putting these slides here, because so many companies consider this as a recording, because whatever I am conducting this training, a copy of this training will be recorded in the YouTube. So they are considering this as an external training record, and they record in the training record as external training. So that is the reason I am sharing the CV, so that they can put the CV, they can take the screenshots, or they can consider the CV as a part of the trainer. Okay, training records. So this is just a brief about me. I have audited more than 500 sites already, I told you, and I am having expertise in many fields. I have faced a lot of audits, so this is what is explained here. If you are interested, you can just pause the video and you can go through it. This is my work experience. I started my career with Sun Pharma. Then I work in a Blue Cross Laboratories. I work in a Alembic Pharma. It was an export-oriented unit. I work in a Zydus Cadila. I work in a compliance department in the Zydus Cadila. I work in US Vitamins, and I work in some multinationals also, which includes AstraZeneca, Sanofi, Mylan. These are my major work experience. And before moving ahead, I just want to put some disclaimer that today the date is 3rd of February, and today we are conducting this training session, 3rd February of 2024. So so today whatever I prepared this presentation, it is in line with the current guideline requirements which is available today. But if you want to implement it, So you need to refer the current guideline. Consider this training, consider this video or the session as a learning session or a training session. If you want to implement, then you have to evaluate it properly, your practices systems, and refer the current guidelines. Okay, that is what the purpose I put in this disclaimer. So you can go through this disclaimer. And now let us discuss our today's discussion topic, that is the continued process verification. And with that, we will start with this, the definition or understanding of a process validation by different guidelines. Okay, so now the point here is, what is the difference between the process validation expectation, you know process validation expectations. So now you can see this, the definition of a UGMP. Even the same definition is available in a WHO guideline, and even the similar type of definitions you will find in the TGA or even the PICS guideline. Okay, so the point here is, when you go through this guideline, you will come to know the key about the definition difference. Okay, why I am discussing this thing, just to make you aware that a different in the definition of a EU and other countries, as well as the USFDA. So USFDA is having something, the different expectations than the other guidelines. So that is what the point I want to clear all of you here. So this is a normal definition by EU or WHO and other guidelines. So what is the definition? Correct, you are aware about this. The documented evidence that the process operated within the established parameters can perform effectively and reproducibly to produce the medicinal product meeting its predetermined specifications and quality attributes. Correct? This is, this definition is clear. Now this definition is expected by so many different regulatory agencies. I referred different guidelines also for that. But here you can see here the documented evidence that the process operated within established parameters can perform effectively and reproducibly to produce medicinal product meeting its predetermined specifications and quality attributes. Okay, now I will put the definition expectation by USFDA, and you can go through this definition. You will come to know the difference. The FDA expectation is, process validation is defined as collection and evaluation of a data. So FDA is focusing on not only to prove that the process is effective and reproducible and all, but how you can evaluate, how you can prove that by collection and evaluation of whole data from process design stage through the commercial production, which establishes scientific evidence that process is capable of consistently delivering the quality product. You got my point. What I want to tell you here is, FDA is more focusing on collection and evaluation of the whole data from process design stage and traditional first 3 batches, whatever you perform the validation, PPQ batches or process performance qualification batches, and continuously in the commercial production batches, so that you will able to establish a scientific evidence and prove that process is capable of consistently delivering the quality product. You got my point here, correct? If you have any questions, please put your questions in the chat box. I will reply to you. But my point is, please understand the key differences between the EU and FDA. And as FDA is having this separate guideline, you can see on the screen the picture of the guideline, first page. So I focus more on the this FDA guideline, because this is more detailed about the continued process verification. Otherwise, if you refer the EU or PICS or WHO or other guidelines, there is only a small paragraph about the continued process verification. So it is more detailed, and all the points covered under by the other guidelines are covered in this guideline. So when you want to implement the continued process verification, you should follow this guideline. You cannot say that you cannot say that you don't have the regulated business or you have only the EU business, Europe business, you don't have the FDA, US FDA business, or you don't have business in US. So why you need to refer the FDA guideline? The reason is, the uh, these requirements are in all the guidelines about the continued process verification requirement, right? So how to implement, what points we should consider, how we should do, that is all detailed in this guideline. So to avoid the non-conformity, you have to refer the FDA guideline. Please understand this point, okay? Because many people will say that, oh, we are not having the business in US, so why we should refer this guideline? So that is what I am again and again explaining you and telling you, that this is the purpose. The continued process verification and even the process validation is more detailed and more elaborated in this guideline. Okay, please, I request all people, please go through this guideline. Okay, so this is what I was telling you about the process validation definitions. And now, this is, you all are aware about that, the process validation is in a three stages. Stage 1 is a process design, stage 2 is a process qualification, and stage 3 is a continued process verification. What does it mean, why I am putting this you all are aware, right? This is, I am not telling any new thing, correct? Only I put in the presentation form. So why I want to tell you is, if you are not implemented, first point is, if you have not implemented the continued process verification, you are not complying the process validation requirements in line with the EUGMP, or in line with the WHO, or in line with the all the other regulatory agencies. So if you don't have the business in other countries also, but as WHO is followed for, WHO guideline is followed for all the export products, all the other countries, you need to comply these continued process verification. You cannot say that, we don't have the business in the other countries, so why it is required? Because WHO is a World Health Organization, you need to follow. And also the all other country guidelines expect that continued process verification should be followed. So if you are not following the continued process verification, that indicates that your process validation is lacking in that, and you have not implemented the process validation approach correctly in your organization. Hope you got my point. Earlier what happened, there was only the process validation means a traditional approach, first 3 batches validation and all, correct? But now the the requirements are something, the different, and that is the reason I want to emphasize here. So stage 1 is process design, stage 2 is a process qualification. Process qualification means simple, you can say a traditional validation approach what you are following now. And stage 3 is a continued process verification, or it is also called as ongoing process verification. So it is ongoing assurance is gained during the routine production that the process remains in a state of control. So this is a stage 3, and that is the reason you should implement continued process verification. Hope you got my point. And now the second point is, if you have implemented the continued process verification, are you complying to the all regulatory expectations? Most of the companies, even I will say that, the all the companies, even whatever I have seen, maybe your company, maybe the, if you have implemented all the things in the last up to last slides, then it is good. But most of the companies I found that it is not implemented in a correct way. So that is what we are going to discuss further in this discussion. Okay, so meantime, if you have any questions, please put your questions in the comment section, chat box. I will take your questions for discussion. Okay, so now the point here is, let us start with the expectations of the FDA. Okay, so a successful validation program depends on information and knowledge from product and process development, correct? So you should not forget about the process development information from the development stage. Okay, the knowledge and understanding is the basis for establishing an approach to control the manufacturing process, so and so, to to get the results of the desired quality products or attributes, whatever it is given in the guideline. But the point here I want to emphasize is the tick marks. What is the expectation behind this? The behind this expectation is understand the sources of variations. Now this is some different concept, variation. What is meant by variation? Variation, I request all people, there may be possibility that we may need to extend some 10-15 minutes this training session. So please bear with me, because this is very important topic. So I don't want to lose your attention. So please bear with me, and I am going to clarify all your points also. So I don't want to just go and read these training sessions or slides. Okay, please bear with me. So manufacturers should understand the sources of variation. So what is my variation? See, when you are considering about the manufacturing aspects, multiple materials are used for manufacturing through the equipment, and then the product is manufactured. So what happens when we are manufacturing the product continuously years together? You gain the experience, you gain the confidence. Correct, I agree with you, I respect that. But when you are taking the materials and you are manufacturing continuously, there will be slight change. There will be slight change continuously will be there in the materials, in the process, in the material manufacturing, in your product manufacturing, in your practices. And these variations, whether they impact on the product quality or not, that we have to evaluate. So understand the sources of variation. What are the possibilities of a variation in our end product? That is what we should identify. Detect the presence and degree of variations. If any variation is there, can we able to detect it? And at what extent the variations will be there, that we should able to detect. Understand the impact of variation on the process and ultimately on product attributes. So if there are some variations, okay, what is its impact? What is its impact on the process which are followed in our company for manufacturing of the end product? Okay, and what is its impact on the product quality or product attributes? And once we identify this, we have to control, we have to put the actions to control the variation in a manner commensurate with the risk it presents to the process and product. These four, you can say, we should aim these 4 points when you want to implement the continued process verification in line with the requirement for process validation. Hope this point is clear to all of you, right? Hope I am not so fast. I am speaking maybe louder to make you understand. And if you feel that I am my speaking is fast, please put in the comment section so that I will speak slowly. Okay, I don't want to take more time, so I want to club all the detailed discussion in a short period. So that is the reason I am doing this. Okay, so if you still require, I will speak slowly. Don't worry, you just put in the comment. Manufacturers should monitor product quality to ensure that a state of control is maintained throughout the product life cycle with the relevant process trends evaluated. This is the expectations by FDA. So now I am discussing about all the FDA expectations first. So what are the FDA expectations? These are all FDA expectations basically. So now manufacturers means what? We. Okay, so now I am considered as a part of the manufacturer. So we should monitor the product quality to ensure that the state of control is maintained. Now you will say that we are monitoring the manufacturing, we are doing some IPQC in process quality checks and all. Wait. Okay, because further it is given. A statistician should create the data collection plans and should also be consulted with regard to the use of statistical method. So what is the expectation is, you need to monitor the product quality, you need to monitor the all the process checks and all. We need to collect all the data, and then that data needs to be evaluated by the statistician. Okay, these are all FDA requirements. Okay, and specifically I put the content in the bold so that you should able to focus on this content. So a statistician should create the data collection plan and should also be consulted with regard to the use of statistical method. Don't worry, I will discuss about the sampling considerations, but these are the requirements. So that is the reason I am focusing these points here. Okay. So further, what are expectations? Statistical tools should be used where appropriate to support any conclusions with regard to the variability and capability of a given process and ensure a state of control. I put directly in the bold is. So you need to apply the statistical tools where it is appropriate, okay, not everywhere. For example, suppose if you feel the impurities. So if you put the impurities in the statistical tools, the very, you will not able to apply it because the impurity limit will be, like not more than 0.1. So you will get 0.05, 0.04, 0.02 below detection limit. So you will not able to put the statistical tool there. So where it is appropriate, you have to apply. Now for example, where it is appropriate, maybe at the stage of yield, at the stage of assay, if it is a dosage form, solid dosage form, at the stage of in process parameters checks like tablet hardness, tablet weight variations, or you need tablet field weight, you know then you can say the tablet the compaction force. In case of injectable, you can consider the volumes, and that way, that type of all the parameters. You got my point? What is its expectation behind this? Differences regarding the subject of sampling in stage 3 of a process validation life cycle, continued or ongoing process verification. It is called as continued process verification or ongoing process verification. Demands a higher number of samples. At least until sufficient data exists to assess the variability. What does it mean? See, it does not mean that you have to collect n number of samples to just go for testing. Somebody will interpret in a different way, okay, that you will perform something, high sampling and go for testing. No, samples means, first of all, you have to get sufficient data to evaluate. So you have to collect more and more data. For example, if you ask me about let us take example of about the the drying temperature, or maybe the temperature operation at the API or in the OSD. Let us say example of oral solid dosage form. So you are applying some temperature. So if you are recording in the batch record, maybe in a 15 minutes or 20 minutes or 10 minutes interval, you will get some limited data. But if you use the data generated and recorded in the PLC of the instrument or machine, that is a programmable logic controller, if you get the data, you will come to know there are so many data points will be there starting to end. So you can use all this data to perform the continued process verification, to perform the variability, to perform and evaluate the variability. You got my point? Second example, if you ask me, like in case of a tablet weight, you are checking the group weight of a tablet. Suppose the group weight you are checking every half an hour interval. So just imagine if the machine is running for suppose 10 hours, you will get a 20 number of data, right? Because every half an hour you are getting the group weight or average weight. So average weight is directly linked to the drug content in the oral dosage form, correct? So if you are taking the double rotary, so you will get about 40 data sets, 40 data points to evaluate the variability in the batch, so that you will come to know in a better way. Or you can get the compaction force. So compaction force is directly linked to the the hardness of the tablets, correct? So how much compaction forces is applied. So each tablet compaction force is recorded in the PLC for compression machines nowadays. You can collect all the data, so that all the tablet compaction force data you are having, you need to collect and you need to evaluate that data to prove that yes, your batch is complying to this requirement. I'm just giving some examples, okay? These are not limited to only this example. I'm just making you aware about what is expected. Of course, sampling also is an important point. Sampling, you need to perform additional sampling also. Don't worry, I will discuss this, but the point here is not only sampling but collection of a data. There I want to put your attention and focus, okay? So what is the FDA recommend is, the FDA recommend that manufacturer use quantitative statistical methods wherever appropriate and feasible. That is what we discuss. And we should not only consider the variability in the batch. Means we have to evaluate intra-batch as well as inter-batch variability. Intra-batch means variability in the only one batch, that is called intra-batch, and inter-batch variability means one batch to another batch. Intra-batch variability means what? In one batch, whether the whole batch is homogeneous, that is one aspect, correct? And inter-batch variability means what? Batch number 1, batch number 2, batch number 3, batch number 100. Among these batches, whether there is a variability or not, that we have to evaluate. And it is a part of comprehensive continued process verification program, and this is the clause given by FDA. Whatever I am discussing, it is in line with the FDA guideline expectations. These are not my words. Again, I am telling you, I am just making for your sake, for your easy understanding. I am making in a simple language. That is only my role is today. Okay. I am just facilitating this training session to make you aware in a easy language. That's it. So hope you got my point. Intra-batch and inter-batch. Why this is the point? Because if you consider the traditional validation approach, what we are doing, 3 batches or maybe 4-5 batches are sampled, some testing is completed, and then protocol and report is prepared, compile the data and report is prepared. That's it. After that, what? After that, you will not able to come to know the inter-batch variation. If there is after validation, if there is a variation in the batch number 7 or 8 or 9, you will not come to know. That is what the point emphasized or focused by FDA. Right? So this is the expectation by the FDA, and further we have to focus on some unplanned deviations. Now, you will say, yeah, we write the unplanned deviations. My dear friends, the unplanned deviations which are detected, you are recording. But you have to put a system to detect all the unplanned deviations or unplanned departure. How? I will give an example so that you will get in a better way. Okay. Let us take the example of, the temperature process only, where temperature is applied. So the temperature you are recording in once in a suppose 20 minutes, and that temperature is applied for about maintained for suppose 5 hours. So every 20 minutes you are checking the temperature and you are recording the batch record. So you come to know that there is no temperature variation. Temperature is maintained within the limit, correct? But you are checking only once in a 20 minutes. In between, if there is a slight excursion or slight change or slight deflection, or you can say a slight the variation, you will not come to know. So how you will come to know? When you will take the data from the PLC of the machine, you will come to know that how many cases, how many points you got the temperature excursion, or how many times the temperature was not maintained, that you will get it. So this is the one of the again the expectation by FDA. The goal of the third validation stage is continual assurance that process remains in a state of control, that is validated state, during the commercial manufacturing. So during commercial manufacturing, you have to put the controls. So what you have to do, you have to put a system or systems. Maybe you need to put one system or multiple system for detecting the unplanned departures from the process as designed in essential to accomplish this goal. So you have to put some system, practices, maybe procedures to detect the unplanned departures which are not detected currently or now. That is the point given by FDA. Okay, whenever I am discussing about the FDA, FDA means USFDA. Okay, so variability now we need to discuss about here. Okay, because we have to identify the variability. So adherence to the CGMP requirement, specifically the collection and evaluation of infrastructure and data about the performance of the process, will allow detection of undesired process variability. So what is meant by process variability? Variability means, simple example I will tell you. Suppose there is a process. If I am manufacturing the process, I will get the product A. If I am following the process, I will get the product A. So this is what you can say the normal process, correct? You got my point? So this is the A, and this is the I will get the product A. But if there is some change, you got my point, this is some change in the raw material or maybe process, minor change, may not able to detect also by you. There are scale minor changes will be there. You remember the variations? In the third or fourth slide we discussed about the variations. Detect the variations, the sources of variations and all, whatever we discussed, remember? So if these variations are there which you are not able to detect, then what happens? If you manufacture the product, normally you get the product A, then you will get something different. Means what? You will get the product A only, but the original product A will have some different properties, while the if in case of variability or variations, and you manufacture the product you will get, it will have some different properties, slightly different properties. That is called the variability. You got my point? So it should allow the detection of undesired process variability. So whatever is not expected, that we should able to detect. Evaluating the performance of the process identifies problems. So when you are evaluating the performance of the process, you should able to identify the problem, and once you identify the problem, you should able to determine whether action plan must be taken to correct, to anticipate and prevent the problems, so that process remains in a control. So when you are performing the continued process verification and you come to know that there is some variability, then you have to able to identify the problem, that why there is a variability, means due to which variation or what type of variations in the equipment, or variation in the operation, or variation in the material, raw material, or variation maybe in the some other aspects is leading to this variability, that you should able to identify. And once you identify it, you should able to correct it, or you should able to anticipate even in advance, and you should able to prevent the problems, so that process remains in control. This is the FDA requirement under this clause. Again I am telling you, whatever I am discussing, it is in line with the FDA requirement. I am just helping you to make you aware and understand in a simple language. That's it. Okay, now up till here you will find, or you will understand that the FDA requirement, requirement of a continued process verification is the collection and evaluation of a data, and we should do this and all. So what people will do, they will prepare a protocol. They will collect the data. The protocol is prepared, some critical process parameter, critical quality attributes, they are collected, and a report is prepared. It is a continued process verification. Answer to this question is no. When we are discussing about the continued process verification, we should have some ongoing program to collect and analyze the product and process data. So you have to analyze the product as well as the process data. That means what? You have to do some additional sampling. Please understand this. That relate to the product quality must be established. So the point is, it is not just a data collection and then you prepare the report. Most of the companies are making the same mistake. What happened? They do the process validation. They traditional process validation, 3 batches or maybe the 10-15 batches, whatever, the 3 batches or 5 batches, I think. They perform the process validation, and after that, as a part of continued process verification, what they do, they prepare some protocol or checklist, and they just fill it, and they feel that the continued process verification is implemented. No, ongoing program you should develop to collect and analyze the product. That means what you have to do, additional sampling, just like validation. Now you have to decide what type of additional sampling you need to do. That you have to decide, okay, based on the nature of the product or property of the product. For example, if it is a controlled drug release, you need to perform some additional dissolution testing during particular continued process verification periodically, or that way, or if it is a small drug content, or the very small molecule, potent molecule, where the drug dose is very small, so you need to perform the the uniformity of dose, or you can say the uniformity of the drug content at the blend stage periodically, not only once during the validation. So that way you have to evaluate. Here, most of the companies fail to comply this requirement. I am telling you, frankly, I am telling you. Hope you're getting my point, what I'm telling you, right? The data collected should include relevant process trends and quality of incoming materials or components in process material and finished product. The data should be statistically trended and reviewed by trained personnel. So the people who are involved in the process validation, they should be trained on that. Statistical trending and evaluation. Hope you remember this. The information collected should verify that quality attributes are being appropriately controlled throughout the process. I am just going through this requirement so that you will understand the things. So FDA specifically recommends that statistician. So now please understand this. This I am not recommending. This is the FDA is recommending, my dear friends. FDA recommend that a statistician or a person with adequate training in statistical process control techniques develop the data collection plan and statistical methods and procedures used in measuring and evaluating the process stability and process capability. So based on the nature of the product, based on the confidence level, based on your data from development to the commercial manufacturing data, the statistician will develop the sample plan, and that data will be collected continuously and evaluated to ensure that process stability and process capability. Hope you are getting my point. Procedures should describe how trending and calculations are to be performed and should guard against overreaction to individual events as well as against failure to detect unintended process variability. There may be possibility that some single event or maybe some one excursion will be there, single or individual events may be there. So you need not take overreaction on that. You need to evaluate why it is happening, why in that particular batch this type of results are there, and you can evaluate based on statistical trend. Okay. Production data should be collected to evaluate process stability and capability. So the production data, just now I was giving some example, that has to be collected, correct? The quality unit, remember the responsibility of quality unit. This is, I'm not telling that that quality unit should do that, but this is FDA is expecting. The quality unit should review this information. Okay, so the production data should be collected to evaluate process stability and capability, and quality unit should review this information. If properly carried out, these efforts can identify variability in the process and or signal potential process improvement. So if you properly perform this, you will able to identify the variability and the sources of variations and all, and you will get some signals also about the process improvement. This is what the FDA is to all of us. Okay. FDA recommend that continued monitoring and sampling of process parameters and quality attributes at the level established during the process qualification stage until sufficient data are available to generate significant variability estimates. So what is mean? Suppose if you are manufacturing the 3 batches for traditional validation batches, so you should not just stop the validation here. You should continue to collect the data and evaluate. You can have some additional samplings in next batches also, unless and until you generate the significant variability estimate or you get the significant data or sufficient data to evaluate that yes, process is under control. You are getting my point? So you should not now stop after validation of a 3 or maybe 4 or 5 batches. You should further monitor the continued next batches. That is what the FDA is expecting. Now you will say that we are having extensive sampling in the validation batches. Should we follow the same for continuing 10, 15, 20 batches? No, 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 no. I am not telling that. Even FDA is also not expecting the same thing. What FDA is expecting that whatever data collection, testing is something different. First, data collection should go on continuously. That is the one thing. Secondly, whatever sampling, additional sampling you are doing, you identify some additional sampling of a validations, and out of the additional samplings, some sampling, additional samplings, you can go in a periodic manner, perform the testing of that additional sampling and develop some sufficient data so as to generate the significant variability estimate. You got my point? Now why these words are used, why these specific numbers are not given, that up to 10 batches you have to do or up to 15 batches you have to do? Why it is not mentioned? Because it depends on your product to product and even the organization to organization, that at what extent the organization is having the confidence or the people are having the confidence on the product. It depends on the type of a product, process design. So many parameters are dependent on that. So that is the reason this type of words are specified. So we have to evaluate based on our process design data, based on our confidence on the PPQ batches, process performance qualification batches, and based on the further nature of the possibilities of the variabilities in our organizations. If you have good automations, good controls, then you will get more and more data in a less batches also. So this is what I just want to share this information about this. So what we are discussing here, about the FDA expectations, FDA expectations. So now this is the slide where the FDA expectation we are going to discuss. After this slide, we will discuss about some sampling considerations, what FDA is expecting about the sampling. And after that, we are going to discuss about the practically what points we should consider. That points I am going to discuss this. So please bear with me, right? So these estimates can provide the basis of establishing level of frequency of routine sampling and monitoring, so and so. Monitoring can be adjusted to a statistically appropriate and repetitive level. This is what FDA has given. These 2 lines you have to put focus here. That is the reason I am not much focusing here. 2 lines. Process variability should be periodically assessed and monitoring adjusted accordingly. Means what? Process variability should be periodically assessed. So periodically, so that means what? Let us say the example that you are manufacturing the new product, correct? So first you will manufacture the normal validation batches. You have the process design data. You have the 3 or maybe 4 or 5 batches you consider for process validation. After that, you monitor the next 6 batch, 7 batch, 8 batch, some 10, 15 batches, and then you can decide some period also, that once in a 10th batch or maybe once in a 5th batch you will further monitor. That way you can make a strategy based on the level of a confidence developed. I am just making in a simple way that how you can implement and how you can understand this expectation. So the process variability should periodically assessed and monitoring adjusted accordingly. And then you need to adjust, So for example, if let us say the example of a OSD product. Okay, I am giving you an example so that make you aware. Okay, this is not a thumb rule. Again, I am telling you, I am just guiding you. This way you can develop. So what happened in normal validation, you perform the blend uniformity, which you are not performing the normal routine batches, correct? You may perform the maybe stratified sampling at the compression stage. which you may not be performing at the commercial normal commercial batches. Then you may be performing the dissolution testing at a different lots of coating lots, which you may not be doing in the commercial batches. In the commercial, you may be performing the dissolution testing of a composite coated samples, not a lot wise, correct? So what you can do, you can adjust this frequency. Means what? Maybe every fifth batch, fifth batch maybe perform the uh, blend uniformity based on again the nature of the product. Okay, again I'm not telling that you have to follow the strain rule, based on nature of the product. So you have to, suppose example, in every fifth batch you perform the blend uniformity. Maybe every 10th batch you perform the drug content. Maybe every 15th batch you perform the dissolution for each lot. So that way you can adjust. My point here is, understand this requirement. You can adjust. It is not a hard and fast copy and paste the protocol for all the products. So the protocol for continuous process verification for one product of the same category of a product, like OSD product, tablet product, suppose, same protocol may not be applicable for the other tablet product. You got my point? That is what my point here is. In normal scenario of a process validation or traditional process validation, most of the time the protocols are similar. Sampling plan, monitoring plan is similar, correct? But here there may be possibility that your monitoring may be different from product to product. That is what I want to emphasize this point here, correct? Hope you are getting my point. Okay, so up to here I was discussing about the FDA expectations and all. Now I am going to discuss about some sampling considerations, because continuously we are discussing about the stratified sampling, sorry, statistical sampling and all. So what are the sampling considerations here? So again I am requesting you, if you have any questions, please put in the questions in the comment section. Good, I am getting a good questions. But my request is, we may need to extend for some period. Okay, because still I think 8 to 10 slides are pending. So please bear with me, because I don't want to skip this any content, so that the content the topic will be limited and all. But as topic requires this, so I need to take some 10-15 minutes extra. So please bear with me. And also one more thing, whenever I will be discussing about the questions and answers, please go through that question and also answer, because from somebody's questions you will get clarity. Okay, so please understand this topic. So what are the sampling considerations expectation by FDA? Statistical sampling procedure in concern with the selection of an individual observation. It helps us to make statistical inference about the population. For example, stratified sampling. So in case you can use the stratified sampling also. If you want to understand more in detail, I will prepare a separate training session on stratified sampling, so that you will understand in a better way. But the point here is, as based on the population and all, even you can have a stratified sampling. Stratified sampling means what? It based on some strata. The group of the peoples are collected, or group of the data is collected, and they are evaluated. That is called the stratified sampling. Simple way, simple wordings. Okay. For example, if you are manufacturing, suppose 100,000 tablets or 100,000 units, so you convert this 100,000 units is something in some groups with respect to the time or with respect to the location. You convert into the groups. That is called strata. That is called strata in the statistical language, and these are evaluated. That is called stratified sampling. The simple meaning I am explaining you. The number of samples should be adequate to provide sufficient statistical confidence of quality, both within a batch and between the batches. Inter-batch and intra-batches. Remember, intra-batch means one batch within the batch, and inter-batch means, intra-batches means what? Within the multiple batches. That we should able to identify. Hope you are getting my point, right? So further, the in-process specifications shall be derived from previous acceptable process average and process variability estimates where possible. So point here is the in-process specifications can be derived, so it may not be fixed. In-process specifications, okay. Now in process specification, if the same specification in the final specification or final quality attribute, then wherever in process specifications are there, they should be below or well within the final quality attribute or final specification. But if that also within the final specification, but that also can be changed based on the previous acceptable process average and process variability estimates where possible, and determined by the application of suitable statistical procedures where appropriate. This requirement is a part establishes the need for manufacturers to analyze the process performance and control batch to batch variability. Batch to batch variability. You are getting my point. This is the the sampling exploration about the specifically the FDA. Now this again, this content I am repeating here. Hope this content you remember this thing, right? But this content purposely I am repeating here. An ongoing program to collect and analyze the product and process data that relate to product quality must be established. You understand this? Now I will give an example, you will understand in a better way. So you have to collect as much of sample as possible. Now I will give simple example so that you will understand in a better way. So normally, suppose if you are performing the blend uniformity testing in a blender, so what you do, you withdraw the sample from different locations, maybe 10 or 11 or 12 locations, whatever, based on the geometry of the blender. But you withdraw the sample in triplicates, correct? So you withdraw the sample in a triplicate sample. So first set, second set and third set. You got my point? Yes. Then what you do? Currently, yeah, most of the company, what they do, one set is given to the quality control for testing, and the remaining two sets, they are kept under the QA control. If there is a problem in the one set, the remaining two sets are issued for investigation purpose. This is the normal practice followed by many companies. But if you refer the FDA expectations, clearly given, to get the more and more data about the drug uniformity, you have to analyze all the samples. Suppose if you are having 10 samples in triplicate, so 10 plus 10 plus 10, that means 30 samples should be tested by quality control laboratory to get more data and evaluate the data. You got my point? This is, I am not telling, my dear friend, this is a FDA guideline expectation. This is specified by FDA. So this strategy is even not followed by many companies, but this is expected by FDA. FDA has clarified this point in a very clear way. Okay. The people who are working or associated with me, they may be aware about this requirement, but I think the people who not work me or not associated closely with me, or the companies where I am not associated, they may not be aware. So that is the reason I am sharing this information to all of you. Okay. So these are some sampling requirements are there. Hope you got the point. And now the main point is how we can implement practically with respect to the continuous process verification. So that is what we are going to discuss now. So variations can also be detected by a timely assessment of. So one aspect about identifying the variation is, we have to assess timely. What we have to assess? The defects. What type of defects we are getting, what type of defects are generated. So when you are segregating the defects, categorizing the defect, that what type of defects are generated, you may able to get the type of variations. Complaints, what type of complaints, OS findings, process deviations, process yields, batch records, incoming raw material records, adverse event reports. This will give you some timely assessment of variations. Further, further, this is not limited to this. I am sharing this. Further, with respect to the changes, what are the plan changes are made? So description of a plan change, a well justified rationale for the change, an implementation plan, quality unit approval before implementation, and it should be documented. Depending on how the proposed change might affect the product quality, additional process design and process qualification activities could be warranted or required that you need to implement. That is also point should be considered when you are practically implementing the continued process verification. So when you are implementing the continued process verification, you need to specify about the what type of change management is followed, what type of change is expected, and what is its impact. That also you should consider. The next point is maintenance of the facility, utilities and equipments. So what you have to consider: qualification status must be maintained through routine monitoring, maintenance and calibration procedures and schedules. So whether you are following the equipment maintenance means periodic maintenance, I am telling, not a breakdown maintenance, but maintenance schedules are followed or not. Any breakdown, then second is the breakdown maintenance, whether after that any breakdown maintenance are that to specify. The equipment and facility qualification data should be assessed periodically to determine whether requalification should be performed and the extent of the requalification. So what is the extent of requalification, at what level requalification should be performed, that need to be evaluated periodically. Maintenance and calibration frequency should be adjusted based on the feedback from these activities. Means if there are some breakdowns are there, breakdown means unplanned maintenance, then definitely the preventive maintenance frequency need to be the reduced, so that you can have a multiple preventive maintenance in a short period and you can avoid the breakdown maintenance, or you can say the unplanned maintenance. So these points you have to consider during the implementation, and this again these 2 points where many companies are lacking, and that also we have to implement these points. So where we are lacking: production line operators and quality unit staff should be encouraged to provide feedback on process performance. Okay, no problem. But here FD recommend that quality unit meet periodically with the production staff. Understand this, I preferably put in the bold. FD recommend that quality unit meet periodically with the production staff to evaluate the data, discuss possible trends or undesirable process variation, and coordinate any correction or follow-up actions by production. So here the point is there should be the periodic meeting with the production staff by the quality units. So questions will be asked during the inspections that how frequently you meet with the production staff and all. So you should be ready with this. The expectations are linked with this expectations by FDA. You got my point? Hope you are getting my point, right? So there are some brief differences between the process validation by EU and FDA. So if you refer the EU guideline, the list of non-critical attributes and parameters in the validation protocols are mentioned, while in FDA specification of critical quality attributes and critical process parameters are specified. The EU approaches are maintained: concurrent, traditional, continuous process verification and hybrid. In FDA there is no distinction. It is very clear: 3-stage process validation, process design, process qualification and continued process verification. It's very clear. Less emphasis on statistical evaluation by EU. In EU guideline, even EU and WHO and other guidelines, there is less emphasis on a statistical evaluation and all. But FDA, as I was discussing, you remember the training session now, more discussion on the statistical evaluation. In FDA, there you need to focus on differences between the subject of sampling in stage 3 of a process validation life cycle, demands high number of samples at least until sufficient data exists to assess the variability. But there is no such a requirement in EU and other guidelines. So that is the reason if you go through the EU guideline or other guidelines, there is only one paragraph or passage So you will not able to know that how I can implement the continued process verification. That is the reason FDA is more clear and more detailed about this. Okay, and after this, there are some warning letters, and as I told you that warning letters, when they are issued, they are not only referred for just only observations and the letters, only the observations and all, but the guidance are also given in the warning letters. The guideline expectations are also given in the warning letters, so that also will be helpful for you as a learning purpose and implementation purpose. So this number is given, from this you can track this. I am not going to read this. If you are interested, you can pause it and you can go through this. I am just sharing this because everything I already discussed this thing. Only I am just presenting this as a part of the learning about the warning letters, that uh, what is expected, that detailed summary of your validation program for ensuring a state of control throughout product life cycle along with the associated procedures. Describe your program for process performance qualification and ongoing monitoring of batch, of both intra-batch and inter-batch variation to ensure a continuing state of control. This way directly FDA has written to the company in the form of warning letter. That means what? The company failed to comply this requirement, and FDA has clearly instructed to do this. That is what my purpose, to put these warning letters as a part of today's learning session. And again, you can go through this slide, that what is expected and all. This is exactly FDA has given in the warning letter. I copied and pasted. You can make a use for implementation purpose from this. Okay, this is all from my side. Thank you. Maybe I speed up the discussions, but I try to cover all the topics and things about this topic. Hope you understand the things. Okay, thank you so much. And now let us discuss about some questions. So I told you that please be with me about the while discussing on the questions, because from somebody's questions also you will learn more things. So first question was asking about the statistical controls and statistical methods. So hope you understand the expectation of a statistical, how the statistician should be involved in developing the sampling plan, and you should able to identify the intra and inter batch variability and all, right. The second question by Mr. Dharmesh is, difference between PQR and CPV. Yeah. Many people will ask this question. We are preparing the product quality review. In the PQR also, we are evaluating the same thing. We are tabulating the raw material data, packaging material data, quality control, quality attributes of the product, then a process parameters, and we are tabulating and we are making the graphical also. So what is the difference in the PQR and CPV? There are so many differences, my dear friend. So first is, product quality review you are performing at the end of the year, but the continued process verification you should perform in between. Not, you should not wait for the year. That is the first thing. Second, you should put some statistical process in the CPV, while in PQR you only focus on the process capability like CP and CPK, but in CPV you focus on process capability as well as process stability. That is the another point where you are focusing here. Third is, in PQR you are only verifying the process variability between batch, one batch to another batch, another batch to another batch, right? Means inter batch. But in CPV you need to focus on intra batch variability. So third thing, fourth thing, in PQR you are only collecting the data, but in CPV you need to collect additional samples periodically. You decide the frequency and you need to test it. Correct? Hope you got my point. Mr. Dharmesh is asking again, in CPV how many batches required for compilation of a CPV report? See, the point is based on what type of products you are manufacturing. So here, instead of discussing on the batches, you need to discuss about the data. How much data you should consider. So as a normal industry practice, about the 20 data points are considered to have a valid statistical estimate. So you can have the statistical evaluation on 10 data points also, but you will not get the valid estimate. So at this minimum, I recommend, it is not given in any guideline, but I recommend from my side at least 20 data points you have to consider. Okay. CPK value required to calculate the CPP and CQ. Yeah, you need to calculate the CPK value for wherever possible. Okay, not all CPP and CQA. Hope you remember the example what I was giving in the uh, impurities test and all. So wherever it is possible, there only you can implement the CPK for a critical process parameter as well as critical quality attribute, right? Then Okay, if you understand, please put that yes, you understood the thing. And some Gussin family UK11 is asking, uh, any all products for process validation. Sorry, your question is not clear. Any all products for process validation, sir? If I understand your question, you are asking about the whether it is applicable for all products. The answer to this question is yes. Why? Yes, yes. All products, whatever we are manufacturing the products, we have to perform the process validation, and continued process verification is a part of the process validation we have to perform for all the products. Hope you understand the point, and if you have any questions, please now you can put in the comment section. Okay, again, thank you so much for your kind attendance. If you are interested in process validation, uh, again other process validation training session, already I have conducted the webinar. I will share the link in the description section. You can go through this link to understand in more better way about the process design and the process qualification, mix stage 1 and stage 2 validation. Okay, thank you so much, and ensure all time compliance. Thank you so much. So if you have any questions, please put your questions in the um, comment sections. I will reply to your all questions. Okay, so as there are no any questions, thank you Mr. Dharmesh for confirming it that you understood this. So if you have any questions, please, I request all people to contact me on comment section. I always reply on every Sunday to your all questions, so that you will get clarity, and on from your questions other team members also can learn. Okay, thank you so much, and ensure all time compliance. I always request all people, all my pharmaceutical colleagues, to ensure all time compliance. Thank you so much. Okay, thank you. And now I am ending this today's training session. Thank you so much, and see you next Saturday same time with the new topic, so that everybody will understand on the different topic. Okay, so see you again next coming Saturday, and on the same time. Okay, so now I am closing this meeting. Thank you so much once again for your attendance. Thank you so much.